Okay, hello, my name is John Robinson. I'm a 2014 Cabrin undergraduate researcher in Dr. Cooper's lab here at the University of Kentucky. And today I'll be giving a presentation over the cholinergic system in Drosophila, and specifically the acetylcholine receptor subtypes. So basically here's the outline of my PowerPoint. There's introduction, methods, discussion, future research, and questions. I'm going to get into the introduction first. The ultimate goal of this project was to determine the acetylcholine receptor subtypes and how they modulate behavior and development in the Drosophila. This gives us a basic understanding of the long and short term under or over expression of acetylcholine or nicotine and its effects of manipulation in behavior and development. We study these because the nicotinic acetylcholine receptors in Drosophila occur in the genome, and they're similar to humans. So we can use the Drosophila as a model organism to study the neural circuitry, because not all pathways are known in both humans and Drosophila. So we can use what we found in the Drosophila experiment as building blocks for humans, and this helps unravel pharmacokinetics, drug pathways, and how transmitters and the effects of certain drugs on the nervous system, how they actually affect humans in the long and short term, is where we're wanting this experiment to take place. What's the main difference between the Drosophila CNS and humans is acetylcholine is a transmitter in the CNS, and is a neurotransmitter in the sensory neurons, but not in the motor, as it is in mammals. So it's similar in structure, but it's different in some of the transmitters. Here you can see a Drosophila stained with GFP, green fluorescent protein markers, and everything lit up in this. I know it's supposed to be green, but it's a black and white photo. So everything is lit up in white is where the cholinergic neurons are. So you can see right here is the VNC, or ventral nerve canal, and then you can see how the neurons extend into the bands of the muscles of the Drosophila, and then how at the head, where the head is, it's really tightly packed, full of sensory neurons. So it kind of just shows you how abundant the cholinergic neurons are in the sensory in the Drosophila. Okay, so the cholinergic system can be broken down into two main parts: the metabotropic muscarinic receptors and the ionotropic nicotinic receptors. The main difference between these two receptor subtypes is that the ionotropic nicotinic receptors form an ion channel and are fast-acting, fast response influxes of ions, and then you get a stimulus outside up here, and then it opens this ion channel, and then you get this massive influx of ions, and it causes a cellular response instantly. However, it doesn't last very long. It's short to act, and it's a short burst, so you get an instant response, but it's not something that will last over time. However, the metabotropic muscarinic receptors relies on a second messenger system. The agonist, such as acetylcholine or muscarine, will bind to a membrane receptor and then activate the G protein, which will affect an effector, and then activate the second messenger, and then you'll get a cellular response. In these, the action isn't very fast, but once it starts, it'll actually form a cascade, so you'll see a longer effect over time. And here we can see we use these because in some of the data we found like a desensitization over time. Okay, and here's a list of the drugs that we used in our experiments and how they're classified as agonist or antagonist. Okay, an agonist is something that promotes a response. They'll actually stimulate like an ion channel or a G protein. So you get a cellular response. And the antagonist is something that will inhibit a cellular response, like close an ion channel, for example. Now, the first 5 drugs are the ones we've already have data for and the ones we tested for. And then the bottom 3, the ones that's highlighted in yellow, are the drugs that we have yet to receive, so we haven't done any actual testing with. And that's where we're wanting to take it to in the future. Okay, the first thing that I want to talk about is heart rates. Okay, in a previous study, they basically did what we did, but they did with an intact animal, which means it wasn't dissected. They just took acetylcholine, for instance, or nicotine, and injected it into the Drosophila larvae. Both the previous experiment and us, we used third instar larvae. They're early stage, so they're not wandering, and late second. So the main difference between us and the previous experiments is that we used a semi-intact model, which means, as you can see in the picture, it's partially dissected, not completely. So it exposes the heart, and I'll get into the anatomy and where everything's located in the next slide. But we do this because the Drosophila heart is myogenic, which means it's not needed, or it doesn't need nerve innervations to beat. So whenever we dissect it like this, we can actually apply the drugs to the heart, and then we won't get a secondary effect. This semi-intact model helps us control all the variables in the experiment. So we get a direct application of the drug and then a direct result thereof. Okay, so in this slide, there's on the left, labeled C, is a Drosophila dissected in the semi-intact method, and it's a picture taken through a microscope. And then the right-hand side, in the picture labeled D, it's an animation of it, and you can see there's 2 sphericals right here. On this one, it's the trachea, and the trachea is actually bound to the heart with ligaments. So if you can't see the heart, then the trachea will actually contract and relax with it. So you can you can see it, or you can get an idea of where it's at, because the heart's actually transparent. So sometimes it's kind of hard to see in the microscope. And this cylindrical tube right here running down the center is the heart, and the little dark spots around it is the pericardial cells. The part of the heart that we use is the true heart, which is located about right here. And then you can see where it starts to pinch off, and that's where the aorta is, which beats a little bit different. So we use the true heart located right here and right here. Right. Okay. What this graph is basically saline exchanges. And we did this because we was noticing we wanted to show that regardless of what the solution is, whenever you exchange solutions in the Drosophila, the heart rate changed. So what we did here was we took 5 points, and you can see them on the graph. And at each point there's 2 saline exchanges, which I'll get into in just a second. And the first point right here labeled number 1 is right after dissection. So in dissection, we put saline in it to kind of help with the dissection. And then it was waited after a minute after dissection was completed, and then the heart rate was counted for a minute. And then at the section 2 right here was actually the first saline exchange. So we exchanged the saline used in the dissection with new saline. Same concentration, same type of saline, comes from the same bottle. It's just taking out the old saline and adding new. And we waited about a minute and then took the heart rates again. And at point 3 right here is 10 minutes after the first saline exchange from 2. And then we took the heart rates, and at point 4 was another saline exchange, and then at 5 was 10 minutes after the second saline exchange. And these in the left graph right here is just the raw averages for each trial. There was 15 in each trial, so these are the averages. There's 5 trials, 15 in each, and then these are the averages for the 5 points. In the right graph is the percent change. So it's a 15% positive 15% change from the initial to the first saline exchange, and then it was a negative 15% change from the first exchange to the first 10-minute mark. And basically what this means is it desensitizes over time. And however, at point 3 to point 4, which was the 10-minute mark, for the first 10-minute mark to the first saline exchange, it actually rose to about positive 15%. And then from the second saline exchange to the last 10-minute mark, at about a negative 25% change. So what we saw overall was it kind of was a negative trend. It was about a 6% change. So basically what we learned from this was once we took the heart rates with the drugs, we would subtract 6% of that, because 6% showed that the heart rate increased or decreased with solution exchanges regardless of what the solution was. So this was kind of to normalize the data. Okay, so right here is the acetylcholine trials. There was 15 used in each of these, and the top graph is the raw averages for the data. So what point 1 is, solution 1 is the initial saline exchange, or was the initial saline from the dissection, and solution 2 is actually the acetylcholine at the first minute. And then we left the drug in for 10 minutes as well, and as you can see in the third point, and the averages were taken for these 3 points and put in. And the bottom graph shows the percent changes in these as well. Solution 1 to solution 2 the first minute, you can kind of see a trend, and then from solution 2 first minute to solution 2 the 10 minute, it's a negative trend. So what we saw was over time the hearts, in my opinion, desensitized to the acetylcholine, and so the heart rates actually dropped as a result. Okay, the next thing that we did was we added an antagonist, which as you remember, inhibits a response. So it should block the heart rate, and then we added an antagonist plus an agonist. So they should counteract one another, which is the basis for this next experiment. And I put this diagram in to kind of show what we did. We put the saline was in first, and then we added the antagonist, and then we added the antagonist plus the agonist. So here's the result. Okay, this is really 2 graphs in one, so kind of bear with me for just a second and I'll explain it. This right here is atropine and scopolamine first minute after the saline was removed from dissection, and this was just the base heart rate, or the base percent change for the heart rate. For instance, atropine, which is a agonist, had a positive 55% change in heart rate. So it increased the heart rate by 55%, and atropine at 10 micromolar increased it even more. And it's kind of puzzling for us because it's supposed to block it, but we're not really for sure why. We're still trying to figure that out. And then in the second graph, right here is after 10 minutes of the initial solution. And you can kind of see atropine is still positive, and even scopolamine in both the graphs are positive. So what we did now was we got new organisms, and then we added the atropine plus nicotine and scopolamine plus muscarine. And what we saw was actually more shocking than what the initial result was, because by itself, nicotine and muscarine increase the heart rate, but with the agonist, it shouldn't do anything. It should decrease the heart rate. But what we saw was actually the opposite effect. It actually increased the heart rate instantly in the first minute, as you can see with the graphs right here. With the atropine, 10 micromolar with the nicotine, and scopolamine plus the muscarine. It's really shocking. And then at the 10-minute mark is basically what we was expecting to see. It actually decreased. So you may be wondering, like, well, what happens? How much does nicotine increase the heart rate? How much does muscarine increase the heart rate? So I added these graphs right here to show the percent changes with the nicotine and muscarine in the 10 micromolar solutions, to show you that it does change it, but it doesn't change it as much as the results, because with the nicotine you get about 75%, and then here you get about 100, all the way up to 175% is what the highest percent change we saw. And then scopolamine's about 90, and then here it's about 100. So it kind of goes down a little, but not much. The next thing we did was behavioral assays, and we started with the body wall movement, and we conducted this experiment by taking 15 late second and early third instars, and then putting them in 0.5 grams of food, and then adding 0.5 milliliters of the drug, so that we get about a 1 gram solution. It's just easy math. But we left them in there for 10, 20, and 24, 10 minutes, 20 minutes, and 24 hours, and to show what the effects of acetylcholine and other drugs would be if once ingested or absorbed through the skin. So to count a body wall, it's a contraction starting at the caudal end, and it moves superior to the head. And as you can see in this little animation, it actually has two, and it looks like a wave moving through the Drosophila. So here you can see in the tail right here, it actually moves up, and then there's another one. See the wave? That's it shows two body wall contractions. So here is the results for the acetylcholine and the 0.1, 0.01, and 0.001 molar concentration. The black bar is the control, which means it was just put in 0.5 grams of food and 0.5 milliliters of water. So it's the same weight as everything else. And then each trial was a different set of organisms. So this trial was independent of this one, and this one was independent of this one. So we're not using the same organisms from the 10-minute to 20-minute to 24-hour feedings. So basically what you can see from here is a slight pattern. You can kind of see the controls a little bit higher than it follows a little pattern in the 10-minute and 20-minute feedings. However, at the 24-hour feedings, both the 0.01 and 0.1 molar increased over time. So the next thing that we studied was the mouth hook movement, which is basically the projection of the mouth hooks of the animal to try to get food, and the retraction, and then that would be counted as one. And what we did here was we used the same feeding technique as we did before, put 0.5 grams of food in a tube, and then added 0.5 milliliters of the drug in it, and then put the larva inside the drug, and then let them wait for an amount of time. And then we put these in a 2% yeast solution, which basically we took a small petri dish, coated the bottom with water, and added yeast to it so the animals would try to eat the food. And then we counted the mouth hook movements in 1 minute. And what we saw here was an increase in 10 minutes, and then a slow desensitization over time of the drugs, except for the 0.001 molar acetylcholine. It's 24-hour. it's a little weird. I don't really know how to explain it other than that. But again, each trial was independent of one another. So we used one set for the 10-minute, one set for the 20-minute, and one set for the 24-hour feedings for the mouth-hook movements. And there's a little trend. You can kind of see a trend here, again, with the 10 and 20-minute feedings. But at the 24-hour feedings, it kind of goes a little crazy. The next thing that we studied was the CNS method. Like I said before, the acetylcholine is a neurotransmitter in the sensory neurons, but not in the motor neurons. So what you have is a circuit, basically. At the sensory, we can stimulate the sensory nerves, and then you'll get an integration in the CNS, and then you'll get a response from the motor neuron. So I'll get into, in the next slide, I'll get into how we did this and the dissection methods and how we actually counted as a result. Okay, so there's three diagrams in here. I'm going to start from A to B and then move to C. A is a fully dissected larva. It's pretty much a fillet. You dissect it, pin it at four corners, and rake out everything. We don't need the hard or the sphericals anymore. We're basically interested in just the nerves. So as you can see in B, it's a better, it's a animation, and it's a better understanding of how we do that, of how we did this. So at the top, you can see the brain, and then you can see the nerve segments coming out. And what we did was we took a suction electrode and took up some posterior roots, and then we stuck an intercellular recording device in muscle 6 or muscle 7, and we would stimulate the posterior roots and then record how the muscles responded. And what these are called EPSPs, or excitatory postsynaptic potentials, because you can only have action potentials in the nervous system, not in the muscles. So we're studying the EPSPs, or the postsynaptic potentials. Once again, this on the slide, the animal was submerged in the drug, so everything is saturated in different concentrations. We used saline as a control, and then we added the drugs accordingly. And then lastly, you can see right here, you can see the stimulus on the bottom. Typically, we stimulated about every 10 seconds, and then you can see where the stimulation stopped about right here. And then each peak is a EPSP, or excitatory postsynaptic potential. And we counted these in set time periods. For instance, let's say 2 seconds. So we'd count from where the stimulation stopped, and then 2 seconds over, and then we would count and then record the data accordingly. So what we have here is 4 concentrations of acetylcholine, and there's 5 trials, 5 different organisms, and 5 readings in each concentration. So we have 25 readings total. And so what these bars are is the percent changes overall in the solution. So there's little percent change in the 100 micromolar, little to none in the 100 micromolar, and there's very little in the 10 micromolar. But in the 100 nanomolar and 1 millimolar, you see a huge percent change, over 100% in the 100 nanomolar, and more than about 200% in the 1 millimolar. And what the dots are is the averages for each trial. So there's 5 dots representing the 5 trials. So the average for 5 runs for this trial is like -5, 10%, and there's it's a 0% change for the next one, and then all the way up at the top, you see a 350% change, and then for the 100 nanomolar, and then for the 1 millimolar, you see a 600% change. If you got rid of these outliers, you can see basically all the other data points overlap. So once the outliers are cast out, everything comes back down, and this really isn't significant because of the overlap. If we saw more scatter in the data, we could call it significant. But based on the trial, based on the percent errors in the recordings, I'd have to say this isn't really significant data, but it just shows you the different results that we got. So in the discussion, we found the heart rates tend to decrease over time, either due to desensitization of the neuron or just something that we're not for sure what's happening. And we found that no matter the concentration of acetylcholine, the heart rates tend to increase with the addition of it. And the mouth and body hook movements showed the same trend for acetylcholine with the 10 and 20 minute feedings, but not for the 24 hours. So from here, what we're wanting to do with the future research is to do the genetic and pharmacological identification of the cholinergic receptors and of the heart, and study the modulatory action, the behavioral and sensory motor circuits and physiology in the Drosophila. And then thirdly, we're looking at the genetic manipulation of the acetylcholine in flies. And we're doing this, we're actually just starting to do this now, and then I can actually show you in the next slide basically how we're going to do this and what's going to happen. So we're using 3 fly lines. CHA Gal4 and UAS GFP is one. UAS Trip line is another, and the UAS Shabira line is the last. So basically what this means is the CHA in the first line is the enhancer for the Gal4, and Gal4 is a transcription factor, and UAS is a binding region, and GFP is a green fluorescence protein, and TRP1 is a protein that will cause acetylcholine to be dumped in the synapse once you heat it to 30 degrees Celsius. And Shabira is actually a protein that will inhibit vesicle fusion, so it blocks acetylcholine once you heat it to 30 degrees Celsius. So in this animation right here, you can actually see the crosses, the CHA Gal4 with the UAS Shabira line, and then in the left side where it says on is the synapse working perfectly, nothing's wrong with it. But what the Shabira does is once heat treated, it will denature a protein, so it keeps the vesicles from fusing, from pinching off and fusing again. So once they fuse, they can't pinch off; they just stay open, which inhibits acetylcholine release. And TRP actually does the opposite. It makes the vesicles pinch off quicker and fill up and dump faster. So you have a massive influx of acetylcholine once it's heated. So with these fly lines, we can see the long and short-term effects of the acetylcholine responses and influxes over time, and see what would happen in the development of a Drosophila, say, without acetylcholine, and what would happen in the development if it doesn't have nearly as much as what it should, or if it has too much of acetylcholine. And there we can see the long and short-term effects of what this will have. But that's basically where we're at right now, and this concludes my presentation. I hope you've all enjoyed it. Again, if you have questions, you can contact Dr. Cooper or Cole Malloy, the graduate student who helped put on these experiments. And I want to thank the Cabrin funding, Dr. Cooper for letting us use his lab, and Cole for teaching us so much. Kyle Ritter is my fellow Cabrin partner here, and Samuel Potter is a researcher in the lab, and so is Sandra. But here's my references, and I thank you all for your time.